good morning, everyone, depending on where you're joining us from. And welcome to today's webinar hosted by Gator Bio in collaboration with Bio, titled NovoBody Platform, Next Generation Antibodies Engineered by Computational Protein Design. I'm Hendrik Lui, Regional Business Manager for Asia Pacific at Gator Bio, and I will be moderating today's webinar. So let's begin. I would now like to introduce our speakers for today. Indrani Chakraborty, Manager of Application at Gator Bio, and Alfredo Chiano Rubio, CSO and co-founder at Bio. Handing it over to our first speaker of the day, Indrani, please.
Thank you. Good morning, good afternoon. My name is Indrani Chakraborty, and I am the Manager of Application and R&D at Gator Bio, and today I will be giving a brief presentation about Gator Bio. Gator Bio, the company, has been founded by Dr. Hong Tang and Robert Zuck. They are the same people who created the original Biolayer interferometry technology. We are headquartered in Palo Alto, California. We do have international locations in Shanghai, China, as well as Basel, Switzerland. Along with that, we have distributors throughout the world. And the mission of our company is to empower researchers by creating a more powerful, productive, and cost-effective BLI system. Gator Bio today currently has 400 plus users all over the world, and our user base keeps expanding with each passing day. We have 60 plus granted patents for our software, hardware, as well as consumables and chemistry. Our users has been generating a whole lot of content and publications. Almost 200 plus publications in leading different journals have been published throughout different groups across all over the world. Our instrument R&D facility is in Shanghai, China, whereas our consumables, which is the biosensors R&D facility, is based out in Palo Alto, California. Instrument manufacturing is in Suzhou, along with some consumables, which is biosensors that are manufactured there. But in Palo Alto, we also have the major biosensor manufacturing facility. Along with all these, we do have a very good team of researchers, scientists who are outbound as well as in-house, every day working to make the Gator Bio a better company in terms of research and development among all its users, current and future. All Gator Bio instruments utilizes a technology called Biolayer interferometry. This technology measures changes in interference pattern that is caused by binding events at the biosensor surface. Now inside the instrument, there is a hardware component, which is the lamp, that shines a white light through the biosensor. And the white light is reflected from the tip and goes into the spectrometer for analysis. The biosensor tip is where the reactions takes place. When the biosensor is in a buffer with no binding event happening at the tip, the sensogram displays a flat line. However, when the biosensor is dipped into a sample well, the analyte binds to the biosensor surface and it changes the interference pattern, resulting in a redshift of the spectrum and a corresponding binding curve on the sensogram. Similarly, during analyte dissociation, the change in interference pattern causes another shift in the spectrum, this time in the opposite direction, generating a dissociation curve on the sensogram. So a key advantage for the Gator instrument, its ability is that it observes binding events in real time, and this enables numerous applications which is not limited to but includes like binding kinetics of antibodies, peptides, and small molecules to its targets, quantitation, epitope binning, cross-reactivity, along with all the aspects of drug discovery. Now from Gator Bio, we have the solution for Gator comes in a package, of course. So it's the machine, which is the first and foremost where the entire experiment or data capture will happen. That is, of course, guided by the software, which is a part of the machine. And there are two components of the software. One is data acquisition software and analysis software, and followed by these consumables or biosensors that has been altered, or they have chemistry on their surfaces in order for the researchers, for them to allow different kinds of experiments or assays to optimize and study in these instruments. So you can get automated quantitation, kinetics, epitope binning, and several other applications. And this machine is very simple, ready to use. As soon as it arrives at a place, you can plug in and start working on it. It's very simple and very fast as well. Currently, the Gator family of instrument comprises of 5 different instruments depending on the throughput and also the sensitivity. The Gator Pilot is the smallest with the least amount of throughput with 4 channel, and Gator Pro has the highest throughput with 32 channel, and the rest in between 8 and 16. Now, along with the hardware machine as well as software, you have what you need in order to do your experiment upon or the surfaces is the biosensors. Currently, we have 35 plus biosensors in different categories that are customized to suit all the requirements. So we have a tag-based biosensor category, where if somebody is using any protein of interest, they are interested in characterizing, measuring affinities or binding constant, or just detecting yes or no binding. Then if it has a his tag, we have ready to use his or nickel NTA tag. So these biosensors comes in pouches where you can open it and it's ready to use. Similarly, if your protein is not tagged, then when you want to see the interaction between its binding pair, if your other component is an antibody, then we have a whole set of antibody specific biosensors, and at any given time you don't need to label any of this in order to see the binding that happens in real time. Similarly, for biotinylated avitag, we have a suite of different streptavidin based biosensor catered towards experimental needs. For today's talk, as we are focused on AI based drug designing that Monad is going to give us, uh, this probably helps in order to understand that how Gator fits in the entire drug discovery process. So, as we see that in every aspect of our life, AI has become so prevalent in drug discovery also in recent years, especially in antibody related improvement of antibody related databases has enabled researchers in order to do drug discovery in silico. But once you have all these molecules and you make them in the dry lab, and then you meet the requirement for a wet lab, because the information regarding affinity, epitope, all these things are missing. So for that wet lab characterization in high throughput approach, especially when you have such large amount of sample volume, these high throughput Gator machines can come into play both in upstream as well as downstream processes. In upstream discovery, you can use it for your hit selection, binding, kinetic assays, epitope recognition, binning studies, etc., or cross reactivity. And when you have figured out what are your leads from this screening process and move on to maybe developability, affinity maturation, at that process also, Gator has always been useful in order for you to develop your drugs and antibody or any other kind of molecules. In order to focus on the most AI-based in silico drug discovery, the two machines that stands out from Gator are Gator Pivot and Gator Pro. Be- the reason being, for the in silico drug discovery, you have such a huge amount of sample. These two machines with 16 and 32 channel can handle this high amount of sample plates. And in some of our users have also actually used Gator Pilot or Pivot and have attached to their liquid handler system, thereby increasing its automation and throughput even more. Now, on top of that, being these two machines being automation ready, also the internal build of the machine gives them a very stable temperature as well as sensitive environment, thereby allowing high sensitive experiments that needs not only high sensitivity but high resolution. Especially for this in silico drug discovery, we are talking about de novo sequence where we have very small size of molecules with a small molecular weight. Here is an example of a kinetic data that we generated using the Gator pivot. Gator pivot, along with keeping a temperature constant, it can also be used in order to do a temperature experiment for different temperatures. Here is an example which we carried out for same binding pair at 20 and 40 degree. And you can see that the differences between the antibody and antigen binding, its parameters are very different when you look at 20 versus 40 degrees. So you can imagine the sensitivity of these instruments and temperature control within the internal instrumentation. Now, not only the instrument is where you need it to be stable in order to give you high sensitive data, but also the biosensor or the surface where the reactions are taking place, it has to have higher resolution or higher sensitivity in order for the user to understand the characterization that is required for these small molecules or small peptides or very small molecular weight entities that are designed during this in silico drug discovery process. And our assay sensor offerings have that capability. Specially mentioned is our SAXT and SMAP, where our SAXT shows almost 300% binding for different molecular weight molecules, starting all the way from a small to a large molecule. And this allows our researchers to carry out a lot of high sensitive experiments for higher resolution. So summarizing all of it, what we can say that Gator Bio is a novel antibody discovery platform, and with its high sensitive, high throughput platform, it enables researchers to characterize small molecules, antibody drugs, or any drug discovery process both at downstream and upstream with its intuitive software and also automation as well as choices of biosensors that it has. Thank you for listening to our webinar, and the next part will follow.
Hello everyone, it is a true pleasure to be here today to talk to you about our NovoBody platform to design next generation antibody mimetics using computational protein design. My name is Alfredo Quijano Rubio and I'm the co-founder and chief scientific officer of NovoBody. A little bit about our company, NovoBody. So we started the company in 2022 with the mission of revolutionizing life sciences tools and diagnostics using AI-powered protein design. We are a spin-off from the Institute for Protein Design in Seattle. We have about 20 full-time employees, big offices and lab space in Seattle, and we have state-of-the-art computational and experimental capabilities. Among our leadership, you'll find experts in protein design and life sciences entrepreneurs, including our co-founder and CEO, Daniel Silva Manzano, our head of business development, Carl Walkie, myself as the chief scientific officer, and also Greg Tibbetts is our fractional CFO. Last but not least, David Baker is one of our co-founders and is one of our scientific advisors. As you know, David Baker received the Nobel Prize for his work on computational protein design in 2024. For this webinar today, I will start with a very brief introduction on computational protein design and will outline some of the state-of-the-art methods used in the field. Then I will talk specifically about the platforms we have developed at NovoBody, and then I will go deeper on our NovoBody platform on designing the novel antibody mimetics. Specifically, I will talk about our latest product, our anti-HISTAC NovoBody, a best-in-class anti-HISTAC reagent. Then, for those of you interested in working with us, I will describe how we can work together, either in a collaboration or by testing some of our existing products. Without further ado, let's talk about protein design. I borrowed some of these slides from the Institute for Protein Design, and I highly recommend, to get a better idea on the status of the field, to watch the novel lectures from last year from David Baker, Demis Hassabis, and John Jumper. As all of you know, proteins carry most of the functions in biology, and there are millions and millions of unique proteins described to exist on Earth. However, this number is only a fraction of the total amount of proteins that could be built by combining the 20 natural amino acids within proteins, let's say from 50 to 1,000 amino acids. And the reason is that evolution hasn't had the time to explore all these possible combinations. Therefore, there's a vast and unexplored universe of possible proteins that now we can explore using computational resources, and that's exactly what computational protein design is doing. So how can we actually design new proteins? What do we need to know? So the first thing we need to know is protein folding. We need to understand how from a DNA we get to a string of amino acids, and how this string of amino acids can fold into a three-dimensional structure that has a given function. This is the problem of protein folding. On the other side, given a desired new protein with a new function that we can come up with, how can we find the sequence of amino acids that encodes this structure, this function, and then actually test it in reality? So this is the problem of protein design. Researchers have been working on protein design and protein folding for decades, developing softwares like Rosetta. But in the last few years, the field has leaped tremendously thanks to developments on AI and deep learning. Here, I put some key publications on protein design and protein structure prediction, especially AlphaFold, that describe some of these key advancements in the field. And it was because of these advancements that David Baker got the Nobel Prize last year for computational protein design, and Demis Hassabis and John Jumper got the Nobel Prize for protein structure prediction. Here, I wanted to briefly show you some of the state-of-the-art methods developed by the Institute for Protein Design in their design pipeline. So first, they use RF diffusion to generate new protein structures, like the example you see on the left, where they generated a new binder for the magenta peptide. Then, they need to find a sequence of amino acids that can encode this new structure, and they use MPNN to find these amino acid sequences. Finally, going back full circle to the problem of protein folding, they use Rosetta Fold or AlphaFold to predict the structure from the sequence, to make sure that the sequence found by the previous two algorithms is accurate and has a high probability of folding and having the desired function. Now, I want to move on to talk about the technologies developed at NovoBody. As I described in the beginning, we are the company pioneering protein design for developing new life sciences reagents and in vitro diagnostics. As you know, traditional research tools and in vitro diagnostics are oftentimes limited because they use proteins available in nature, including monoclonal antibodies. These tools can be expensive to develop, insufficiently stable, lack sensitivity, lack characterization, and are difficult to manufacture. So what if we could use the power of computational protein design to enhance these existing tools or even create new ones that can be even better reagents to accelerate research and make it more efficient and cheaper? So that's exactly what we're doing at NovoBody. We're building new proteins using AI-powered computational protein design technology to create tunable tools that are rapid to develop and integrate seamlessly with existing reagents and existing platforms. Since we launched the company in 2022, we have created multiple platforms that deliver products. So if you actually go to our website, you can see that we have multiple products available for purchase, but also we're happy to talk to develop products together with you. So we spun out in 2022. The first platform that we developed was Luxit Pro. Luxit Pro is a de novo luciferase. Luciferases are luminescent enzymes that can be used as reporters and are ideal for detection assays, cell assays for drug discovery. Luxit Pro is the first de novo enzyme that has key properties, is the smallest luciferase, the most stable, and one of the brightest. And we have developed a full luciferase and a split form of the luciferase. This product is available on our website. I'm happy to discuss more after this webinar. Then we developed the NovoBody platform. The NovoBody platform is going to be the center of the talk today, but it's basically our platform to develop antibody mimetics. We are using proprietary AI protein design algorithms that deliver highly optimized de novo binding proteins that are oftentimes superior to monoclonal antibodies. And the best of all is that we can deliver results within weeks. The third platform that we developed is the NovoLysa platform. This is a fast and sensitive one-step biosensor kit that can be used for research use or in vitro diagnostics. And basically how it works is that we have the NovoBodies that bind to an analyte of interest, and then once bound, they activate our split luciferase, LUX8, to generate light in proportional amounts to the amount of analyte present in the sample. And this happens within 5 minutes, so it's very quick. We have one of these products on the website, so you're welcome to take a look. I'm happy to discuss more about this after the webinar. Finally, our latest platform is the protein mimetic platform. So basically, given an existing enzyme or functional protein that has some liabilities or limitation, we can take this and apply our computational design methods to enhance their properties while keeping their activity or improving it to really generate best-in-class assets. As I said, today, the NovoBody platform will be the focus of our talk, but happy to discuss any of the other tools after the webinar if you want. That being said, let's move on to the focus of the talk today, which is the NovoBody platform using AI to create a new antibody mimetics. I assume that all of you have used antibodies either for research or diagnostics at some point, and all of you probably are aware of the good, the bad, and the ugly about antibodies, right? They can be expensive to develop, insufficiently stable, lack sensitivity, lack validation, take a long time to optimize, and are also difficult and expensive to manufacture. So our proposal was, can we build on top of the decades of knowledge and developments in antibody engineering, but upgrading them with computational protein design to find proteins, find sequences that don't exist that can be better in some applications than antibodies. So basically, there are untapped solutions in the protein design space that we can access with our NovoBody platform to create new products that can be superior for different applications. So our NovoBody platform is our proprietary AI de novo binder design platform. Basically, we incorporate the best of monoclonal antibodies and upgrade them with protein design. The idea here is that we can incorporate binding information from existing antibodies, scFvs, or other binding molecules like nanobodies to create a NovoBody, which is a high affinity, high specificity molecule that is small, is only about 29 kilodaltons, single chain, hyperstable, has no disulfides, has no postulational modifications, and can be expressed in both E. coli or mammalian cells. And is fully customizable. So you can customize properties like isoelectric point, tags of interest, chemical handles of interest, the location of them, and I will explain more about this later. So basically what you can enable here is, instead of having a limited assay because of the limitations of your antibody or your reagents, now you can customize the reagent to fit the needs of your assays, which is something that the feedback we get is that has been impossible up until now. So what this means for users is that it reduces using NovoBodies reduces cost of reagent and assay development, can enhance assay performance because now you have optimized reagents that fit your assay, really to create best-in-class reagents and assays. On top of that, we'll talk more about this later, but it makes very simple the creation of bispecific binders. And the best of all, this happens in weeks. We don't need to do a discovery campaign. We don't need to create molecules from scratch. We can do this development from conception of a NovoBody, from an existing antibody, to a vial of manufactured protein that you can test in your lab within 2 weeks. It's a very, very fast process. So the next question we get all the time is like, okay, have you validated this platform? The answer is yes, and extensively. So we have developed more than 75 unique NovoBodies, and that bind diverse molecules like proteins, peptides, small molecules, and phosphorylations. All these binders have high affinity and specificity, and as you can see on the graphs, most of them have picomolar affinities or single-digit nanomolars and have very good cross-reactivity. I think it's important to mention on this slide that we do all the measurements using a Biolayer interferometry instrument, particularly the Gator instrument, and that's why we're here on this seminar. And I will discuss more in detail later how we use them for screening. Expanding more on the NovoBodies that we've built. So these include some for in vitro diagnostic use. Many of our NovoBodies bind cardiac markers, oncology markers, inflammation markers, or infectious disease markers. But also we have many of them for research use only. We have NovoBodies for flow cytometry, drug discovery, cell manufacturing, protein manufacturing, or quantitative assays. In addition to the ones that we have developed internally, we can also do custom projects where a partner comes with an idea or an antibody that they want to change format to a NovoBody or optimize. And we've taken plenty of these projects, and we can deliver materials within 4 weeks. And the best of all is that we believe this is just the beginning. So in silico, we have designed more than 2,500 NovoBodies, and we just need time to test them all because we're a small company. But we can see a future where we have NovoBodies available for any existing biomarker. So how do we screen our NovoBodies? So starting from the sequence of an existing monoclonal, we use our proprietary AI platform to create sequences that encode the NovoBodies with the binding properties that we want. Then we order the synthetic DNA. We order a handful of candidates, and then we express them in E. coli and test them for soluble expression and binding using a Gator instrument. So within 7 days, we can find our designed lead NovoBody candidate that then we move on to manufacturing. Next, I want to share with you a specific example of a NovoBody we developed recently and that we launched commercially only a few weeks ago, and that is gaining a lot of traction with customers in academia and companies. This is our anti-HISTAG NovoBody. For this product, we wanted to take on a difficult challenge. So as all of you know, anti-HISTAG antibodies are infamous in the field because they have very low affinity and low specificity and don't perform really well in many applications. So our challenge was, can we take a well-known antibody that binds HISTAG with very low affinity in the micromolar range, make a NovoBody, and then optimize it in a unique way that we have not seen with antibodies before to make a reagent with the highest affinity possible. So that's exactly what we did. So we took the parental antibody, made a NovoBody, and measured its affinity. As expected, because we didn't do any in silico maturation, the affinity was similar to the parental antibody, micromolar. Then we did 4 rounds of affinity maturation and improvements based on our knowledge, and we got to a NovoBody that had a sub-nanomolar affinity and performs really, really well. As far as we know, this is the anti-HISTAG reagent in the market with the highest affinity. So here we highlight some of the properties that make this reagent very exciting as an alternative to existing commercial antibodies. So on the left, you have Biolayer interferometry assays run with a Gator system that compare in blue our NovoBody with 2 of the most cited antibodies in the market. One of them has nanomolar affinity, the other has micromolar affinity, and as you can see in blue, ours has picomolar affinity. Even beyond that, since the NovoBodies are much smaller, you can also see much more signal and resolution on the BLI instrument. So actually these molecules are a very exciting alternative to traditional antibodies to be used in Biolayer interferometry. In addition, another key and superior property of NovoBodies is that they are more stable than antibodies. So what you see on the right is a thermal recovery assay where we are comparing the resistance of an antibody and the NovoBody to different cycles of increase and decrease in temperature. And what you're seeing in red, the antibody, after multiple cycles, it starts aggregating and it doesn't go back. So basically it loses its function. However, in blue, the NovoBody maintains structure, doesn't aggregate, and can resist multiple cycles of temperature, really highlighting that these molecules are resistant to temperature changes, but also are generally more stable than antibodies. One key application of our anti-HISTAG NovoBodies is flow cytometry. Traditional anti-HISTAG antibodies have been challenging and unreliable to use in flow cytometry because of their low affinity. However, our HISTAG NovoBody, because of the high affinity, performs really well in flow cytometry. Here you have one example on our biotin-related NovoBody detecting a HISTAG protein in flow cytometry. We have plenty more examples on our website. You're welcome to take a look. Another promising application of NovoBodies is their use for protein purification. Here, we conjugated our anti-HISTAG NovoBody to an agarose resin, and we compared its use for purification of HISTAG proteins to a classic nickel NTA resin. On the left, what you can see is that actually the protein coming out of the column is much more pure than traditional resins. On top of that, this resin can be regenerated multiple times. What we did here was multiple cycles of binding to a protein of interest, elution, regeneration, binding, elution, regeneration, up to 40 times. And as you can see, we did not lose performance of the resin. This is enabled by the high stability of NovoBodies and its reversibility. If you're interested in checking out what we have available, please go to our website where you can find multiple options, including modifications to the NovoBody like biotin and FITC, or different tags like a twin strep tag or human FC. However, if you have a particular example or customization you want to develop, please let us know, because we can easily modify and customize these reagents. One of the key properties of NovoBodies, as I mentioned earlier in the presentation, is that they can be fully customized for your assay of interest. And this is because they are designed from scratch, they are small, highly stable, and have no post-relational modifications. Some examples are that we can customize the isoelectric point, the size, but also the amino acid composition. And what this enables is something really cool. So for example, since NovoBodies are designed without any cysteines or disulfide bridges, we can then add one single free cysteine far away from the CDRs to use it for maleimide-based chemistry, to conjugate enzymes, fluorophores, gold nanoparticles, nucleic acids, or even conjugate to surfaces in a site-specific manner that doesn't affect the function of the NovoBody. What this enables, as you can imagine, is much better precision in the conjugation, but also a better performance of the assay. Even beyond that, we can do something very unique that it's impossible with antibodies. So for those of you that want to use amine reactive chemistry instead of maleimide chemistry, we can also create site-specific conjugation methods. And the way we do it is that, as you know, antibodies have lysines spread all over the surface of the structure. And NovoBodies can be designed with lysines also on the surface of the protein, but we can design the NovoBodies without any lysines. Then we can couple them with a synthetic tag, a de novo tag that we have designed in-house that has a given number of lysines. This can enable is amine reactive site-specific chemistry that is far away from the binding interface so that it doesn't affect the binding properties, and also allows for providing the conjugated molecule with a given geometry and orientation within an assay. So this is a very unique feature of our platform. Furthermore, some customers want to take advantage of the unique properties in affinity maturation of the NovoBodies to obtain, like we did on the HISTAG case, something with very unique affinity and specificity, but then go back to a full antibody. So of course we can do this by two methods. First, we can just create humanized antibodies, but also we can just make an Fc fusion and we can fully customize these molecules for your needs. I also want to talk very briefly about the power of the NovoBody Duo platform. The NovoBody Duo, as the name indicates, is a combination of two NovoBodies. And this is a very simple thing to do because we can genetically fuse them together. This is a very powerful platform to create bivalent molecules, biparatopic molecules that bind to two different epitopes on the same molecule, or bispecifics. A quick example here showing how this improves the performance, the binding affinity of the NovoBodies as measured in Biolayer interferometry. So we can see how a monovalent NovoBody binds with nanomolar affinity, but then when we make it bivalent, it increases the avidity and then we get a sub-nanomolar binder. The same applies for a monoparatopic NovoBody that binds to one epitope on analyte of interest. When we fuse it to another NovoBody that binds to a different epitope, we can see a massive increase in on rate, but also decrease in off rate, really achieving remarkable affinities. Additionally, making bispecifics is really simple. Doesn't require any chain exchange like with antibodies. It's a very simple fusion. And as you can see here, the NovoBodies binding to their own targets, when connected together, fused together to make a bispecific NovoBody duo, they retain the affinity for their molecule of interest. So here I want to take a moment to show you the list of NovoBodies that we have in development. So we have dozens of antibodies that bind biomarkers important in vitro diagnostics on the left, and also multiple NovoBodies that are useful in multiple biochemistry assays for example, protein purification and flow cytometry on the right. So please take a look, and these products are in development. They are not on our website yet, but we're really happy to work with you to do an early evaluation of these new soon-to-be products. And that's a perfect segue on how to work with us. So if you're interested on any of our platforms that include our de novo luciferase Luxi Pro, our de novo antibody mimetics, de novo bodies, or our de novo fast immunoassays, NovoLysa, you can either reach out and we can provide a quote and then you can purchase them. We can work on a material transfer agreement with you, work on a distribution agreement, or do a licensing agreement. We have many options on the table and we're happy to discuss with you. Additionally, in cases where you want to develop a new product that is not on our list, please reach out because we are always happy to do a research collaboration agreement with you to develop unique products in collaboration. So please let me know in the questions after the webinar if you have any projects that you want to start. And with that, I want to thank everyone for your time. Please reach out to our email that you have on the slide, or please reach out to me directly. We are really happy to hear about any ideas for new products that you would like to see soon in our catalog and available for purchase, but also happy to discuss new projects or new exciting, unique applications that you have in mind. So please let me know. Thank you very much.
Okay, thank you very much, Alfredo and Indrani. We're now going to begin answering the questions submitted during today's presentation. So as a reminder, to all participants, you can still submit questions right now through the Q&A window pane on your control panel. So our first question here is for Alfredo. All right, so Alfredo, is NovoBody similar to scFv?
Right. So it is actually very different. So how we design the NovoBodies is, as I explained, using AI to create new scaffolds. So the sequences are very, very different than traditional scFvs if of either species. Like it's not similar to human, mouse, or any other species. Yeah, but structurally can sometimes can look a little bit close to what an scFv would look like. But we make sure to optimize them to basically optimize the stability and the function as I explained.
Okay, great. Thank you very much, Alfredo. We have a second question here, probably for Indrani. So Indrani, can you explain to us what are the key advantages of using BLI over SPR for biomolecular interaction analysis?
Yes, Hendrik. So, of course, BLI and SPR both are label free, and both of them has its unique advantages. Just to compare and contrast, what I like as a user of both of the instrument that I have used extensively. So BLI is very easy, simple, plug and play. You need very minimal to almost no training in order to get your hands on and get going to run experiment on the platform. Whereas SPR, it is complicated. You need to learn how to operate the machine, analyze and run assays. So it will take time for you to get used to even use SPR. So if you can imagine that in your lab, if you have BLI, it's like your workhorse. It has no fluidics involved, so thereby you can use BLI for any kind of sample usage, which you are not afraid of clogging your microfluidics at all at any given time. So the downtime for your machine is absolutely none for BLI compared to SPR. And another thing is for BLI platform, you have a lot of ready-to-use biosensors that is not available for SPR platform, where you have to make the chips and then you start your experiment. These are ready to use. You can open packages and start using and testing them. Also, the samples that you use, which are very precious, of course, in BLI, it's there in the 96 well plate and you can reuse those samples later. But for SPR, because they have injection that goes through the flow cell, so you ran out of the sample. You cannot recover those samples. So these are, and sensitivity wise, of course, the SPR is little more sensitive compared to BLI. I can just think of both, having to use both side by side depending on what is your need. So both has its unique advantages and maybe slight disadvantages. If that answers your question, Hendrik.
Great, thank you very much, Indrani. That's an excellent response. Okay, so this is for Alfredo. All right. Do NovoBodies contain artificial sequences?
Yes, so we use AI to design the sequences, and again, with the objective of making them the most stable and better performing. So yeah, the sequences are actually artificial. I think I mentioned it on the previous question. If you align our NovoBodies with existing antibodies, they are quite different.
Yes, great. So there's a follow-up question to that. So aren't antibodies with artificial sequences problematic as human drugs, especially in terms of I think he's referring to immunogenicity.
Right, that's a great question. So, and it has a very complex answer, and maybe we don't have the full answer here. But I guess there are multiple aspects to immunogenicity. So, when designing a drug, we can certainly include a step during our design process. We're actually avoiding any known to be immunogenic MHC 1 or 2 peptides, right? So that's a step that we can do in silico to avoid any hypothetical problems with the drug if we're designing drugs. But then stability is a big factor, right? So actually it's known that unstable proteins are much more immunogenic. So that's actually an aspect that the novel proteins generally would have an advantage over traditional native proteins. But when we compare the peptide composition for our de novo proteins versus others, like an scFv, for example, we see that they're very similar because you can design for that. Now, for every drug, you need to test the immunogenicity, and that's something that needs to get done before putting them in the clinic for sure.
Okay, so well, I think you probably kind of answered part of this third part of this question. So, are you envisioning the development of diagnostic reagents using the NovoBodies, or rather than antibody drugs, as antibody drugs? Yeah.
Yeah, and happy to clarify that. So, as I explained at the beginning of the conversation, that's really the mission of Monod Bio. I will say this is not our first company. So our executive team we have worked in therapeutics in the past, but for Monod, we saw a big opportunity to really start creating reagents for users like everyone in this seminar that can really improve your research, accelerate, get more precise, and just do better research to develop whatever you're developing. So that's what we're doing. And actually, the great thing about this is that you don't have to worry about immunogenicity. So you can just focus on creating the best tool possible.
Okay. So next question is for Indrani. Are there any differences between the Octet and the Gator BLI? What are the advantages of the Gator BLI?
Okay, so there are certain advantages of the Gator BLI versus the Octet BLI. Of course, it's in the hardware that is kind of not very visible compared to the users who use it day in and day out in terms of the lamp and the groove and things like that. But what is very obvious for the users who use it on day to day is, of course, the amount of the ready to use biosensors that Gator Bio has to offer in terms of choices. As I mentioned during the talk, like we have quite 35 plus, and also we have custom biosensors that is not listed, and we work with our users in order to make ready to use biosensors for them. Along with that, I wanted to mention that last year, I believe there was a publication called Biolayer Interferometry and its application in drug discovery and development in analytical chemistry, where they discussed in detail about the similarity and differences between Gator and Octet. So I think the questionnaire, I think his name is Akash. He can take a look at it, or I can send him the link for that particular paper that also puts it side by side.
Great, thank you very much, Indrani. Okay, next question for Alfredo. So can you design NovoBody from scratch without using any existing monoclonal antibody as an initial input?
That's also a very good question. And as you probably know, it's one of the hottest topics in the field, right? Like many companies, including us, are working on this. And we've been, like when I say we, is at the Institute for Protein Design, but also at Monod, we've been successful in a few cases. We at Monod, since we prioritize developing reagents fast and efficiently so that we can have like a big number of molecules that people can use quickly. So we found that actually starting from like the anti-His, right? Starting from an existing antibody that is underperforming and not working really well, and then applying our expertise in computational protein design, but also experimental development, we found those challenges to be really the sweet spot for us, because we can create something much better than what exists in a much faster timeline when it takes weeks or a few months instead of like many months to years, right? As with some challenging projects where you're trying to create de novo antibodies.
Okay, thank you very much, Alfredo. There's one more question for you. So how do we so I believe he's this participant is asking, so how can they exercise an RCA with you to develop a novel body of an existing antibody?
Oh, I love that question. We can provide our email contact. It was on the slides, but we can definitely provide it later on email. But they can reach out if they Google my name. They can find my email online or my LinkedIn. So feel free to contact me directly, and we can connect.
Okay, great. Thank you very much for that. Okay, so maybe maybe I think we have time for a couple more questions. So for Indrani, someone is asking what kind of consumables are needed to run experiments on the Gator system, and how frequently do they need to be replaced?
Sorry. So consumables, all you need is biosensors And depends on what type of assay you are developing or carrying out. Most of our biosensors are regenerable, and you can reuse them at least 10 plus times, so that gives you like a lot of benefit in terms of able to reuse. Now suppose you are doing an antibody antigen binding experiment, then your antibody, if it's a human or a mouse, depending on what you would buy, a human FC biosensor or a mouse FC biosensor, and that can only be it. You don't need any others, because you can capture your antibody and now bind the antigen to see the direct binding interaction, and you can keep regenerating the the biosensors over and over again. So we tend to make most of our biosensors regenerable, except the streptavidin, of course. The streptavidin biotin bond is very hard to break, but we do have a flex assay where we have regenerable streptavidin biosensors. I hope that answers the question.
Great. Yeah, that's a great answer. Right, Alfredo, back to you again. What is the purity of the analyte and whether you can use the cell extract expressing the target protein directly for your binding assays?
Yeah, that's a great question, and actually, it could be both for us or for me or for Indrani. But I can answer. Yes, we use the, as Indrani said, we use the Gator as our workhorse in the lab. I think it's used more than 8 hours every day, continuously testing new proteins, and and it works really well when you use directly extracts. So that's a very easy way that we have to, go directly from a design to, in a few days, test how well it binds to the target of interest.
Okay, great. Yep. So that really is going to be very useful, right? To be able to use analytes or ligands directly from the cell extract. And then one last question for Alfredo: do you have de novo design NovoBodies?
Yeah, this relates to the previous question. So we have a few in the pipeline. We haven't announced them yet, but we are definitely working on that. But we also have many other that are, a hybrid in what I said, basically starting from something that it's suboptimal and then optimizing it with our expertise. So we have basically many approaches to designing new proteins. So if anyone's interested on anything in particular, happy to talk, because we usually have at least 3 or 4 approaches that we can take to design the best possible proteins with the properties someone wants, and making them fully customizable.
Okay, great. All right, I think we are pretty much done with questions. Thank you very much, everyone. So thank you to our speakers, Alfredo and Adriani, and thank you everyone for attending today's webinar. So if you have any other questions, feel free to contact us at info@gatorbio.com. All right, so on behalf of Monop Bio, Gator Bio, and our presenters today, thank you once again for joining us, and have a great rest of the day.